Today we'll present about COVID-19 and why it's important to stay home. So what is COVID-19? COVID-19 is an enveloped, single-stranded, positive sense RNA virus. It has the potential to spread from species to species, animals to humans. COVID-19 belongs to a family of coronavirus. On the right-hand side, you can see the symptoms listed by the CDC, such as fever and shortness of breath. And below are some preventions, such as frequently washing your hands and wearing a mask, highlighting the importance of hygiene. Research is important right now because of the uncertainty of the virus and allows us to find the possible treatments.
Here are two important biochemical applications used in research. One is RT-PCR, which is real-time polymerase chain reaction, where viral RNA is amplified and expressed in DNA. With this technique, we can detect the presence of viral proteins. A positive results in the fluorescence of a sample obtained from a patient. Another antibody testing technique you can use is enzyme-linked immunoabsorbent assay, known as ELISA. ELISA is where the virus antigen is used to bind to potential antibodies. This interaction can be observed with spectrophotometry. Through the ELISA, we can use to find potential antibodies present from patients who have recovered from coronavirus and use that antibodies as a source for treatment for further pandemics.
To unravel the disease, it would be essential to combine ELISA and PCR techniques. As the name suggests, PCR ELISA combines an immunological method to detect and quantify specific PCR products directly after the mobilization of DNA. Some future testing, like contact tracing, sewer testing, and antibody analysis, must also be taken into account, as they would enhance effective methods to approach COVID-19. For the most part, although COVID-19 is not well understood yet, we believe that taking into consideration the information provided can enhance a foundation to combat COVID-19 in the near future.
And the takeaway is to remember to stay home. So on our second infograph, we're exploring Chlorella sorkiana's potential and the new key it is into the future. Algae can be used for lipid fuels, food sources, and bioremediation. The main purpose of this experiment that we conducted was to culture and isolate the sample of CS and assess its growing condition and its overall application. On the left-hand side, you can see images of our bioreactor where we cultivate and isolate CS. The main point here is that the cultivation of CS involves light, oxygen pumped into the bioreactor, some nutrients such as nitrogen and phosphorus.
On the right-hand side, you can see the graph that shows the nutritions were depleted around day 5. The growth of the algae started to decrease. The blue line represents the absorbance at 680 nanometers, while the red line represents the absorbance at 750 nanometers. The bottom two bar graphs show that the nitrogen phosphate consumption. On the bottom left shows the graphs of the cell count versus the absorbance.
From our pistachio water experiment, we've yielded a biomass of 0.3519 grams. In comparison to another experiment who used the same organisms but different restrictive factors, growth factors, they've obtained a biomass of 0.1 grams. Based on our, we hypothesized that due to the restrictive and competitive environment in which these microalgal cells are in, it induces emergency storage components of lipids to fuel their biological needs with limited resources of phosphate and nitrate.
Without any doubt, this experiment has increased our understanding of algae while linking the phenomenal roles it plays in life. The established benefits of algae in the near future are its usage in purifying water, a sustainable food source, and in relation to this experiment, provide a potential resource for energy, reducing pollution, and enhancing a better world for everyone.